ladies and gentlemen, you know, being biochemists and microbiologists, I'm going to speak about molecular mechanisms of magic peptides discovered and developed by Dr. Harrison. So you've heard enough that these peptides for sure induces different physiological processes. They improve the physiological status of the cell, of the organism, and they actually prolongate the lifespan. So there is no doubt. But what about molecular mechanisms of these particular things? So the peptides, sorry, again something. Okay, the peptides which we use, indicated here in a short list, you will see. Of course, in the institute of Dr. Hamilton, this synthesized plenty of different peptides, but we use these particular peptides, consisting of 3 to 4 amino acid residues. Okay, so I'll give you only a few examples of these particular peptides, what they're really doing in the cell, how they do interact with the cell, and what they are doing with the cellular structures, with DNA, with transcription, and so on, with the most important unit genetic functions. So let's consider a little bit about pancreatin. So you may see here that pancreatin induces the expression of very important differentiating factor as a PAX-C protein being introduced into the cells. As a matter of fact, we've been first on the world to show that the DNA methylation itself has got aging specificity, so it changes with the age. And besides, we have established that DNA methylation is a tissue-specific or cell-specific. And we propose, and just written quite easily and clearly, that DNA methylation is the mechanism of regulation of transcription as well of cellular differentiation. It was just a first in the world indications of the importance of the important DNA methylation itself, enzymatic DNA methylation. You know, at that time nobody believed that in eukaryotes DNA methylation may play some essential physiological role. But nevertheless, we just continue. How's to work? I don't know how to put it here. Oops, maybe this one. No, doesn't work. No tricks. Something wrong. Okay. No tricks, please. No circus tricks. Okay, therefore, I've been interested to learn what's happened when the cells are no again. It's not happening. Okay, so something wrong with the system. Okay, we just try to see what's happening with DNA methylation, especially the DNA methylation in promoter gene area, because you know methylation of promoter is responsible for genes to be active or not. You see here, that's here. You see here on this left panel is CPG sites methylation. It's a target sites to be methylated in the genome. So in these pancreatic cells, those contain a couple of the CPG sites which are methylated. But on the right, panels representing the excrements when the cells were grown in the presence of the pancreatin, this specific tetrapeptide. So you may see here. Look at the down, it's very difficult to find it, the down on the right panel. all CpG methylated in promoter area are disappeared. So they somehow demethylated in the presence and under the influence of this particular peptide. Another example, when another gene promoter was methylation started, it's also the left-hand side, it's the control cells, panels, and the right-hand side, the experimental, when the cells were drawn in the presence of peptide. Again, you see that the CpG methylation disappeared when the cells were grown in the presence of this particular peptide. So this is a very important thing. What about another peptide? It was bronchogen, which was important to be investigated. So you may see here that from this particular peptide increase essentially transcription of the early genes differentiation factors in the human epithelium, lung epithelium cells. The same happened with the late differentiation factors, these genes indicated in the right side. So the asterisk just stays for the significant distinguishing things associated with the increase in transcription of these particular genes. So look, these two peptides do influence the transcription of specific genes, specific tissue-specific platform. Again, you may see here that in the presence of this particular peptide, this one, methylation of promoter area of genes disappeared. Very strong influence. So, taking all the things into consideration, we suggested a hypothesis with Dr. Hanson, indicating at least that it was just the hypothesis showing the model of how peptide may influence the expression of the genes. According to our suggestions, peptide may bind specifically to promoter area, and this binding protect this area to be methylated by respective DNA methyltransferases, and the promoter piece to the left being unmethylated. But we know for sure, for 99% of genes, the demethylated promoter is obligatory thing for the transcription. So, we have observed, and Dr. Hanson partially told in his report, that these magic peptides, they work not only in human being cells, but they're commonly working in different other organisms, and you may see here how peptides, different bronchogen and epithelium, induce the growth of the tuberculous tissue. So we have looked for the expression of specific growth regulation factors in these particular plants. You may see here again that the peptides specifically increase the transcription of growth regulation factors in plants also. This is very interesting observation. We have observed that peptides, they do modulate the expression of genes responsible for the synthesis of the indigenous, more long peptides, which are known as the plant hormones. So this is a quite interesting event, because the small indigenous peptide may regulate the expression of long peptides in the genus. So it's very important and interesting observation. Nevertheless, besides this may testify to existence of common mechanisms of peptide regulation in eukaryotes in general. When these peptides originated, whether this system originated of short peptides regulation of gene expression, nobody knows, but they still do exist, it does exist in the organism of different systematic opposition, different in evolution, and of course it should originated in the early steps of evolution. Okay, this is just a fact indicating that they are very important. They're doing some sort of the revolts in the physiology of different cells. But what about about the mechanisms of the action? You have seen shortly this line, but you know, the question is whether these peptides are able to penetrate not only into the cell, but into the nucleus. So we incubated HeLa cells in the presence of fluorescently labeled peptides, and you may see here, look to the right-hand side, that fluorescence is clearly observed in the nucleus. And not only in the nucleus, look at the total, over the total nucleus, but it's observed even in nucleolus. So there is no doubt that on the way to the nucleus, peptides used may meet and probably interact with very different structures of cytoplasm as well as the nucleus, including this proteins, cRNA or different RNAs and DNA itself. Another interesting observation that peptides, this peptide may modulate the action of specific enzymes recognizing DNA sequences, which are maybe methylated or not. So this is very badly impossible to use this. Well, you can't see, and me also. So on the left you see that just DNA in the gel. And then 3 empty spaces. This DNA is hydrolyzed completely by our specific enzyme and the nuclease, and the other, you may see that in the presence of epithelon, hydrolysis is partially blocked. Okay, wonderful also. Completely blocked. So, you know, the binding of peptide to the DNA in specific sequence inhibit the action of restriction enzymes, but perhaps it may inhibit also the action of the respective proteins interacting with DNA also. So this is showing you how fluorescence decreased when DNA was titrated with peptides. No doubt that peptide may interact in the full DNA. And also we have some specific sequences, oligonucleotides containing the sequences CC, which are target sites to be methylated. And the addition of epithelon, again you may see decrease the fluorescence. So it means that peptides may recognize specific sequences, including DNA methylated sequences. Very important to be clear here that peptides distinguish between sequences which are methylated or not methylated is very important. I'd like to remind you, sorry about it, the main mechanisms of epigenetic actions in the cell. First of all, enzymatic DNA methylation. Second, I call it as a secondary genetic code. And then enzymatic histone modifications. You see, it's called a histone code, and then the genes are in this siRNA. So let's see what's happened with histones. This is just a picture of sketch of the chromatin structure. H1 histone is located in the internucleosomal area, and core histones are inside of the nucleosomes here. So here is just DNA is laying, not staying, but laying, and you know the histone tails, they're outside of this complex. And then these tails are going to be modified by specific histone modifying enzymes. This modification may be very, very different, indicated here. So we have isolated histones purified, and we got individual histones, H1, 3, and so on, and obtained the fluorescent level histones. By titration of these fluorescent histones with peptides, in all cases we see how the fluorescent decreases. It means that peptides are able to interact not only with DNA, not only with peptides, not only with deoxyribonucleosides, still at that time. So, but also they do interact with the histones. Very important. They interact here not only with H1 fractions of histones, but also with the core histones, H2, and so very important observations. And the binding properties are different. It's depending on the histone nature and primary structure of peptides itself. Okay, short peptides modulate, as we have seen, methylation of the DNA, and therefore, like DNA methylation itself, they may control all specifically all genetic functions in the same cell, in the different cells. No, including DNA replication, transcription, DNA repair, and so on. Peptide activation of the transcription is associated often with CpG site demethylation in promoter area. And finally, specific peptide bonding with histones may be another control mechanism of genetic functions. Thus, short peptides, which I was talking about, short peptides represent here the efficient signaling system of epigenetic control of the cell physiology. The further investigation of these peptides, of course, is very important to elucidate the molecular mechanisms of gene transcription and molecular mechanisms of evolution itself. And concluding, I'd like to say that search for and design of new short peptides, biologically active peptides, is a key promising way to origin and production of a new generation of drugs that are gene addressed and strongly needed to prevent premature aging and to treat cancer and other different diseases. Thank you.
Thank you. One short break.